Gas exchange. Respiratory exchange of gases take place at two sites, namely first in the respiratory surface or respiratory organ and then in the cells or tissues. The former take place between the O2 of the surrounding medium and the CO2 of the blood or body fluid, and the latter between O2 of the extracellular fluid and the CO2 produced within the cell. Mechanism of gas exchange. Respiratory gas exchange is purely a physical process without energy expenditure. It takes place by simple diffusion across the respiratory surface. For high rate of diffusion and maximum intake of O2, most organisms continuously move air or water over their respiratory surface. This is called ventilation. Flow of water out of the gills in aquatic animals and breathing in and out of air in terrestrial animals are examples of ventilation. Then factors influencing gas exchange. The rate of respiratory gas exchange is influenced by several factors. The important ones among them are the following: pressure gradient or tension gradient of the gas, solubility coefficient of the gas in the liquid medium, diffusion coefficient and diffusion capacity of the gas in relation to the cross-sectional area, distance and viscosity of the liquid. Then molecular weight of the gas, ventilation perfusion ratio, temperature, thickness and surface area of the respiratory membrane. The rate of gas exchange in general is directly proportional to the temperature and surface area of the respiratory membrane and inversely proportional to the molecular weight of the gases and the thickness of the respiratory membrane. Pressure gradient or tension gradient of gases. Gas exchange takes place by simple diffusion in a pressure gradient or tension gradient from a region of greater partial pressure or greater tension to a region of lower pressure or lower tension. Partial pressure is the pressure of an individual gas in a gas mixture. Tension is the pressure of a gas in a liquid. In mammals, the partial pressure of O2, that is PO2, is always greater in the inspired alveolar air and the tension of CO2 is greater in the capillary blood. So O2 diffuses to blood from alveoli and CO2 diffuses from blood to alveoli. In tissues, the tension of O2 is always greater in blood and the tension of CO2 is greater in tissue fluid. So O2 diffuses to tissue fluid and CO2 diffuses to blood. Next, solubility coefficient of gases in the liquid medium. Solubility coefficient is the quantity of a gas absorbed by 1 ml of liquid at a temperature of 0 degrees Celsius and a pressure of 760 mmHg. The tension of a gas in a liquid is directly proportional to the volume of the dissolved gas and inversely proportional to its solubility coefficient. The volume of the dissolved gas in turn depends on pressure and solubility constant. Volume of dissolved gas is equal to pressure into solubility coefficient. Tension of dissolved gas, volume of gas in the liquid into 760 by solubility coefficient at 760 mmHg. The tension of gases is measured by the instrument tonometer. Diffusion coefficient and diffusion capacity. The diffusion coefficient of a gas is the volume of the gas in 1 ml of a liquid diffusing over a surface area of 1 square centimeter and along a distance of 0.001 mm. Diffusion capacity is the volume of the gas diffusion across the respiratory membrane every minute in a pressure gradient of 1 mmHg. The diffusion capacity of the membrane for oxygen is 21 ml per minute. The diffusion capacity for CO2 is nearly 20 times greater than that of O2. Diffusion capacity is directly proportional to the cross-sectional area of the membrane and the solubility of the gas in liquid medium, but inversely proportional to distance. Then ventilation perfusion ratio. Gas exchange in the lungs depends markedly on a ratio between the air flow, ventilation and blood flow, perfusion into the lungs. The ratio between the alveolar ventilation and alveolar vascularization is called ventilation perfusion ratio. Gas exchange is most efficient when each alveolus of the lungs is fully ventilated and fully perfused or fully supplied with blood. Abnormalities in this ratio are the most common causes of hypoxia and they often result from a wide range of cardiopulmonary disorders. Normally, alveolar ventilation rate is 4.5 to 5.04 per minute, and alveolar vascularization rate or cardiac output is 5.04 per minute. So the overall ventilation perfusion ratio is 0.9 to 1.0. Alveoli with ratio significantly below 0.9 are relatively over perfused, and those with ratios above 1 are relatively over ventilated. In over perfusion, the excess blood flow is not fully oxygenated and is therefore considered to be shunted blood. The excess blood flow that lowers the ventilation perfusion ratio is called physiological shunt. Similarly, in overventilation, the excess ventilation does not significantly increase the oxygenation of blood. Thus, the excess ventilation is wasted, and so it is considered to be added or alveolar dead space. Alveolar dead space. In alveolar ventilation is zero ventilation, but blood flow continues. Gas exchange ceases. So also when the ventilation perfusion ratio is below normal, the lung will act simply as an inert gas exchanger. Alveolar ventilation will become inadequate for blood flow. Less CO2 than normal will be removed from alveoli, and the pCO2 in the alveoli will rise. Rise in alveolar pCO2 will decrease the PO2. Decreased PO2 in turn will reduce the O2 content of the blood, leading to arterial hypoxia. When alveolar ventilation exceeds blood flow, pCO2 will decrease and PO2 will increase. And adaptations for gas exchange. Gas exchange is a simple process in aerobic bacteria and small and simple aquatic organisms such as protists, sponges, cnidarians, flatworms, etc. Oxygen from the surrounding medium directly diffuses into the cell, and the CO2 diffuses out from the cell. So specialized respiratory structures are needed. However, in larger and complex animals, a direct gas exchange is inadequate or rather impossible. This is because most of their cells are not directly exposed to the air or water that surrounds the body. So such animals require specialized and richly vascularized respiratory structures and surfaces. In large animals, the surface area provided by these structures for gas exchange far exceeds the area of general body surface. This is to meet their increased oxygen demand. Human lungs, for example, provide a total surface area that is nearly 40 times greater than that of the total skin area. In the respiratory surface, blood flows in close proximity with the external medium. This facilitates gas exchange between them by simple diffusion across the respiratory membrane. The respiratory surface is always thin and moist and is formed of richly vascular epithelium. In land animals, the respiratory surface is always kept moist by glandular secretions to enable the diffusion of gases. The principal respiratory structures found among animals include the body surface or skin, branchiae or gills, tracheae, book lungs or lung books, then book gills or gill books, pulmonary sacs and true lungs. Accordingly, 4 kinds of external respiration can be recognized among aerobic animals. They are cutaneous or integumentary respiration through skin, branchial respiration through gills, tracheal respiration through trachea, and pulmonary respiration through pulmonary sacs and lungs, which are the cutaneous, branchial, tracheal, and pulmonary. First, let's learn about cutaneous respiration. This is the gas exchange through the skin or body surface. It occurs in most annelids, some mollusks, some arthropods, and a few vertebrates, amphibians, and some fishes such as Angula, Periophthalmus, then Boleophthalmus, etc. The skin of these animals is thin, moist, glandular, richly vascular, and easily permeable to gases. Significantly, most of these animals have a low metabolic rate, so only a small quantity of oxygen is needed. Cutaneous respiration is considered to be the simplest and the most primitive type of respiration. Then branchial or gill respiration. Branchial respiration or gill respiration is the respiration with the help of gill books and gills. Gill books are found in lemurus, and gills are common among aquatic animals such as annelids, mollusks, many arthropods, echinoderms, primitive chordates, fishes, and fully aquatic amphibians. Then tracheal respiration. Tracheae are the respiratory organs of insects, centipedes, millipedes, some mites, and some spiders. They are thin-walled, branching and interconnecting air tubes. Tracheal system communicate with the outside through valvular openings called spiracles or stigmata. First, let's learn about cutaneous respiration. This is skin through skin or body surface. Then gill respiration. Gill books or book gills. Book gills are found in lemurs. Then tracheal respiration. Trachea are the respiratory organs of insects, centipedes, millipedes, some mites, and some spiders. They are thin-walled, branching and interconnecting air tubes. Tracheal system communicates with the outside through valvular openings called spiracles or stigmata. They are for the entry and exit of air. Tracheal tubes end in blood-filled branches called tracheoles. Tracheoles are the sites of gas exchange between the O2 in the tracheal fluid and CO2 in the body fluid. Hemolymph. In tracheal respiration, blood has no role in gas transport. So air enters the blood through spiracles. It is then distributed all over the body by the tracheal system. Blood has no role in gas transport. And last one, pulmonary respiration. Pulmonary respiration is the respiration by book lungs, pulmonary sacs, and true lungs. Book lungs are found in scorpions. A book lung consists of 15 to 20 million vascular plates arranged 15 to 20 vascular plates arranged like the leaves of a book. Pulmonary sacs are air-filled mantle sacs found in amphibious and terrestrial mollusks like pila, slugs, snails, etc. True lungs are found in lungfishes, amphibians, reptiles, birds, and mammals. Lungfishes are significant in that they have gills for aquatic respiration and lungs for aerial respiration. Let's learn about respiratory pigments. Respiratory pigments are complex colored and metal-containing conjugated proteins involved in the uptake, storage, and transport of respiratory gases. They have very great affinity for O2. A high PO2, they loosely and reversibly bind with O2 to form unstable and easily dissociable compounds. At low PO2, these unstable compounds dissociate, releasing O2. The oxygen content of the blood of most animals is greatly increased by the combination of oxygen with respiratory pigment. So, in the absence of respiratory pigment, the oxygen content of the blood would be very small. Antarctic ice fish is an exception among vertebrates in that its blood lacks respiratory pigment. So, the oxygen uptake by its blood is very low. The absence of respiratory pigment in this fish is compensated by increased blood volume and higher cardiac output. The commonest respiratory pigments among pigments include hemoglobin, myoglobin, hemocyanin, hemoerythrin, then chlorochronin, echinochronin, and echinochrome. Of this, hemoglobin is found in both the vertebrates and invertebrates, and others are found only in invertebrates. Hemoerythrin, chlorochronin, echinochronin, and echinochrome. Vertebrates use hemoglobin as a circulating respiratory pigment and myoglobin as a storage respiratory pigment. Hemoglobin is an iron-containing and oxygen-carrying red pigment. It is found in the erythrocytes of all vertebrates and in the blood plasma of some vertebrates like ascaris, some flatworms, several annelids, arthropods, and mollusks, and sea cucumbers. The protozoans, Paramecium and Tetrahymena, have been shown to contain hemoglobin. A modified type of hemoglobin called leg hemoglobin is found in the root nodules of leguminous plants. The normal hemoglobin found in adult human beings is called hemoglobin A, HbA, and that a fetus is called fetal hemoglobin or HbF. HbF has higher affinity for O2 than HbA has. It is synthesized in the fetus before birth. It may be almost completely replaced by HbA by the 12th month after birth. So adult human beings contain hemoglobin A, HbA, and that a fetus is called fetal hemoglobin or HbF. HbF has higher affinity for O2 than HbA has. That is how the baby is present in the womb get oxygen. It is synthesized in the fetus before birth. It may be almost completely replaced by HbA by the 12th month after birth. Hemoglobin is a conjugated metalloprotein. It is formed of a colorless and globular basic protein called globin. It is an iron-containing portion or prosthetic group called heme, H-A-E-M, heme, H-E-M-E, or hematin. Hematin is the oxidized form of heme. Heme is an active group taking part in O2 transport. Hemoglobin varies with species. Even in the same individual animal, there may be different kinds of hemoglobin. All the different kinds of hemoglobin have the same heme group but different globins. So they differ from each other only with regard to their globin part. Molecular organization of hemoglobin. A human hemoglobin A molecule consists of one globin molecule and 4 heme molecules. A heme group is attached to the globin through a coordinated histidine nitrogen atom. The globin molecule in turn is a tetramer formed of 4 polypeptide chains, namely 2 alpha chains and 2 beta chains, A2, B2. Alpha 2, beta 2. In HbA, the 2 alpha chains and 2 gamma chains in HbF. Okay. These chains remain linked with each other and also with the heme molecule through weak non-covalent bonds such as van der Waals forces, hydrogen bonds, salt linkages, and hydrophobic interactions. Each alpha chain is formed of 141 amino acid units and each beta, delta or gamma chain is formed of 146 amino acid units. The molecular weight of human HbA is around 66 to 684 daltons. Heme molecule is an iron porphyrin complex. It is formed of an iron atom and a divalent porphyrin molecule, a naturally occurring pigment which is a derivative of porphyrin. Iron occurs in the ferrous state, Fe2 plus state. The porphyrin unit is tetrapyrrole ring. Tetrapyrrole ring. It is formed of 4 pyrrole rings with centrally located ferrous atom. The porphyrin of hemoglobin bears a vinyl group CH double bond CH2 on one of the 4 pyrrole rings. In chlorochronin, vinyl group is replaced by methyl group CHO. Now it becomes clear that a hemoglobin HbA molecule consists of 4 amino acids, 4 polypeptide chains, 4 iron atoms, 4 porphyrin molecules, and a vinyl group. Hemoglobin has an active role in the transport of O2 and CO2. The most important property of hemoglobin is its affinity for O2 and its ability to reversibly bind with oxygen. The hemoglobin molecule can bind with as many as 4 oxygen molecules. Its affinity for O2 depends on temperature, pH, PO2 and PCO2 of the blood and the concentration of 2,3-diphosphoglycerate 2,3-DPG in the blood. 2,3-DPG. The ability of Hb to bind O2 enables it to serve as an oxygen carrier of blood. This reaction is unique in that the ion of the heme continues to remain in the ferrous Fe2 plus state, forming a oxyhemoglobin complex HbO2. Oxygen only loosely and reversibly combines with the ferrous ion without actually oxidizing it into ferric ion. So the iron atom gets only oxygenated and not oxidized. Oxygenated, not oxidized. The oxidation of iron is prevented by a cover of the hydrophobic groups of the globin. The oxidized ferric ion has no oxygen carrying capacity. The oxidized ferric ion has no oxygen carrying capacity, and so it is biologically inactive. A bare oxygen free hemoglobin is called deoxyhemoglobin. In adult human beings, there is yet another type of adult hemoglobin called hemoglobin A2, HbA2. Its globin part is formed of 2 alpha chains and 2 delta chains. Hemoglobin HbA2, hemoglobin A2, HbA2. Each alpha chain is formed of 145 amino acids and each delta chain is formed of 146 amino acid residues. Alpha 2, delta 2. The hemoglobin content of the blood of a normal human adult consists of about 97% of HbA, 2% of HbA2, and 1% HbF. The normal level of HbA in blood is 14 to 16 gram per dl in male and 13 to 15 gram per dl in female. Abnormal hemoglobins or variants of hemoglobin. Apart from normal hemoglobins HbA and HbF, some abnormal hemoglobins are also found in human beings. They include sickle cell hemoglobin or hemoglobin S, HbS, Cooley's hemoglobin or hemoglobin C, HbC, hemoglobin D, hemoglobin E, HbE. Sickle cell hemoglobin HbS is an abnormal hemoglobin found in sickle cell anemia patients. It differs from HbA in having an amino acid valine instead of glutamic acid in the fifth position of its beta chains. Hemoglobin C, HbC or Cooley's hemoglobin is the abnormal hemoglobin found in thalassemia, Cooley's anemia patients, thalassemia. In it, normal glutamic acid in the fifth position of the beta chain is replaced by lysine. Hemoglobin D results from the replacement of beta 121 glutamic acid by glutamine. Hemoglobin E is a highly prevalent variant of Hb. It results from the replacement of beta 26 glutamic acid by lysine. It is common among the Orientals of Southeast Asia, Bangladesh, Myanmar, Thailand, West Bengal, etc. Hemoglobin D. Then myoglobin. Myoglobin, commonly called the muscle hemoglobin, is somewhat similar to hemoglobin. However, it is a monomeric globular protein formed of only one ferrous atom, one heme group, iron porphyrin group, and a single polypeptide chain of 153 amino acids. Its molecular weight is 16,700. So myoglobin is found in skeletal and cardiac muscles. Its affinity for O2 is much greater than that of hemoglobin. So it may get saturated with O2 even at very low PO2. Functionally, myoglobin serves as an intracellular storer and carrier of oxygen. It releases its oxygen store only when the PO2 is very low, as in strenuous muscular activity. At times of oxygen deficiency in muscles, it may release its oxygen store to be used for metabolic purposes. Then hemocyanin. Hemocyanin is a copper-containing pigment. It is found dissolved in the blood plasma of a few arthropods like lemurs, some crustaceans, and some arachnids, and some mollusks, cryptochitin, then vesicon, sepia. Its molecular weight ranges from 1 million to 7 million. It is colorless when deoxygenated and blue when oxygenated. A hemocyanin molecule is formed of 2 copper atoms linked to a polypeptide chain. It differs from hemoglobin in that a porphyrin ring and the heme group are altogether absent in it. Hemocyanin take part in O2 transport. A hemocyanin molecule can bind only with one molecule of oxygen. It is colorless in the deoxygenated state and blue colored when oxygenated. In some mollusks, blood contains hemocyanin and muscles contain hemoglobin. Thus, the presence of one respiratory pigment does not exclude the presence of other. In some animals, hemoglobin or hemocyanin may be replaced or supplemented by a minor pigment. Such pigments include hemoerythrin, chlorochronin, echinochrome, etc. Echinochrome is a red-brown pigment found in echinochromes. So contrasts between hemoglobin and hemocyanin, you could see hemoglobin, porphyrin ring and heme component present, but in hemocyanin, porphyrin ring and heme component absent. Hemoglobin metallic part is iron, metallic part is copper in case of hemocyanin. Then hemoglobin red when oxygenated and deoxygenated, blue when oxygenated and colorless when deoxygenated in case of hemocyanin. Then hemoglobin remains bound with RBC in chordates and dissolved in blood plasma in some non-chordates. Hemocyanin remains dissolved in blood plasma in some arthropods and mollusks. One molecule of Hb can transport as many as 4 molecules of O2 at a time. One molecule of hemocyanin can transport only 1 molecule of O2. And we try to learn about all this in a quick glance. Try to learn about hemoglobin. Red colored iron porphyrin protein carried in plasma in non-chordates or in RBC. So hemoglobin is a red colored iron porphyrin protein carried in plasma in non-chordates or in RBC chordates. Distribution in paramecium, some parasitic flatworms, ascaris, some annelids, renicola, some arthropods like Daphnia, Chironomus, some mollusks, then sea cucumbers, chordates, etc. Hemocyanin, copper-containing protein, carried in solution in plasma, blue when oxygenated and colorless when deoxygenated. For example, in crabs, lobsters, prawns, chitins, cephalopods, gastropods, and arachnids. Then hemerythrin, iron-containing protein, carried in coelomocytes, purple when oxygenated and brown when deoxygenated. Spongulates, then brachypods, polychaetes, etc. Then chlorochronin, iron porphyrin protein, carried in solution in plasma, green colored one. Some families of marine polychaetes are there. Hemerythrin is iron-containing respiratory pigment found in some polychaetes. For example, Magellona, and spongulates, then brachiopods. Its molecular weight is around 100,000. It is purple when oxygenated and brownish when deoxygenated. Hemerythrin is contained in some wandering cells called coelomocytes, which circulate in the coelomic fluid. A hemerythrin molecule contains several iron atoms without a porphyrin group. One oxygen molecule combines with 2 or 3 iron atoms. Chlorochronin. It is an iron-containing green-colored respiratory pigment found in the blood plasma of marine polychaetes. Then Serpullidae. It is green both in oxygenated and deoxygenated states. Its molecular weight is about 3 million. The structure of chlorochronin is similar to that of hemoglobin.